Hello my dear learners, welcome to Learn Microbiology. In this another video on cell cycle, we will be studying about the negative regulators of cell cycle that are your PRB, P53 and P21 proteins. Let's get started. First, let us understand what is cell cycle regulators. I hope by now you all know what is a cell cycle, and your cell cycle comprises of G1, S, G2, which are called as the interphase, and the M phase where actual mitosis takes place. In the previous video, we also studied about the cell cycle checkpoints. Over there, we learned that there are three major checkpoints of the cell cycle, namely your G1S checkpoint, which is between the transition of your cell cycle from G1 phase to the S phase until and unless the environment is favorable and your DNA is ready to undergo replication, the cell cannot move further this checkpoint. The second checkpoint, which was known as your G2M checkpoint, it was between the transition between your G2 phase and your M phase. Then we had another checkpoint which was called as the spindle checkpoint, which was present at the transition between your metaphase to anaphase, and we studied over there that unless and until your chromosomes are properly attached to the spindle fibers and they are aligned at the metaphase properly, anaphase cannot take place. I hope that is clear. So let's now move ahead and understand about the regulators of the cell cycle. Cell cycle regulators are of two types. One is a positive regulator and second one is a negative regulator. Positive regulators are those molecules when they are activated, they will initiate the cell cycle or they will allow the cell cycle to progress further. While negative regulators are those protein molecules when they get activated, they will halt or they will stop or they will block the cell cycle. So we have learnt about an important positive regulator that are cyclin CDK complexes. Yes students, we learned that when cyclin CDK molecules combine together and they get activated by phosphorylation, they allow the cell to progress from one phase to the next phase. Now, what are these negative regulators? The examples of it are protein RB, we have P53 and we have P21. So, in this video, we are going to study the three important negative regulators that is having the function to halt or to block the cell cycle. Now students, why this is important? This is important because we studied that unless and until the particular events that has to take place in a particular cell cycle is finished or completed, the cell cannot move to the next phase, right? So for this we require these negative regulators which will stop your cell cycle until and unless the events are favorable for the progression of your cell cycle to the next phase. Before moving ahead, I just want to let you all know what this negative regulator proteins, these are also called as the tumor suppressor proteins. They are encoded by your tumor suppressor genes. Now what is so? These particular negative regulators have been first discovered in tumor cells because we know tumor cells have lost the control over their cell cycle and they are now going to divide uncontrollably. So it is understood that these particular three proteins which used to halt the cell cycle are not halting the cell cycle, means there is a mutation in these genes, and because of the mutation in these particular genes, tumor has resulted. Hence, these genes are called as tumor suppressor genes, and if they get mutated, they will lead to tumor or cancer. Let's move ahead first with our discussion about the PRB protein, which is called as the retinoblastoma protein. This protein, when mutated, was known to cause a tumor called as retinoblastoma, which is a tumor of the retina. Okay, so hence the name retinoblastoma protein. Now, let us understand in detail about the function of this retinoblastoma protein. Before that, you have to keep one thing in mind, that when this protein gets activated, this will stop or this will block the cell cycle, and that is the one which is beneficial for a cell cycle. These negative regulators, even though they block the cell cycle, but the eventual aim or their goal is that irregular or improper cell cycle is prevented. So how this particular retinoblastoma protein works is, we have one particular molecule which is called as E2F. Now this E2F is a transcription factor. Now what is a transcription factor? Transcription factors are those molecules, they will go and bind to your promoter region, and that will allow your RNA polymerase to bind also to the promoter region. It is basically going to align your RNA polymerase in a better manner on the promoter region so that the transcription of the downstream genes will take place. So this E2F protein, this is known to bind to this promoter region, and when it binds to this promoter region, RNA polymerase can bind here and it will allow the transcription of cyclin A gene. Cyclin A plays a very important role in your S phase, and it is going to bind to your CDK2, and both of them together are called as your SCDK. It means that this particular gene, if it is transcribed, it is going to code for your cyclin A molecule which will bind to CDK, and they play a very important role in S phase. Thus we can say, if this E2F goes and binds to the promoter, then the cell will progress to the S phase. Now what is the function of this PRB now? PRB is going to hold your E2F. Now this PRB is an active protein. Now it is activated. So once this PRB is activated, it will go and bind to your E2F, and it will prevent the E2F from binding to the promoter and transcribing the cyclin A gene. Now until when this is going to happen? Until when PRB receives a particular signal which says that now everything is okay, now cell can proceed to the S phase. Only then this PRB gets inactivated and it can no longer bind to your E2F. Let's see how it happens here. Now one of the signal is cell grow in size. When the cell grow in size, then the cell becomes ready for replication. What happens is, we have your PRB still bound to E2F. We know in our G1 phase there are two particular CDK molecules. One is your DCDK4 or 6 is having here a activator phosphate. So this molecule is an active molecule. Cell is in the G1 phase, and this particular G1 cyclin is active and it has done its function. Now one of the important function of your G1 cyclin is to add this particular phosphate to PRB. Students, do you remember we studied over there that CDK kinases are those enzymes, when they are active they will transfer the phosphate to a target protein. So here this PRB is one of this target protein. So G1 phase cyclin has come here and it has told that everything is okay, and thereby it phosphorylated your PRB. Now this PRB has just got one phosphate. So it is now hypophosphorylated and still it is active. To inactivate it further, we have one more CDK complex, that is your ECDK2, that is called as your G1S cyclin. Now this G1S cyclin is again going to transfer its phosphate to this molecule, and now we can see over here our PRB molecule got phosphorylated and now it became inactive. So once it became inactive, it can no longer have a hold to your E2F and your E2F is released. Now when E2F is released, what it is going to do, we already discussed. It is going to bind to the promoter region. RNA polymerase is going to bind over there, and that is going to transcribe your cyclin A gene. Now once transcription done, then we have translation and you have your mature cyclin A protein ready. Now who is going to bind to cyclin A? It is your CDK2 molecule, and they combine to form together your ACDK2. Now this ACDK2 molecule is, once it gets activated, it can allow your cell to progress to your S phase. Students, I hope you understood the function of PRB. This is a very important topic concerning entrance exams. Let's revise once again. PRB holds your E2F together, and that will not allow your transcription factor E2F to bind to the promoter and transcribe the cyclin A gene. If so, the cell will proceed to the S phase. So until and unless your PRB gets signal that cell is ready to go to S phase, it will hold your E2F with itself. But once it gets the signal that now the cell can move towards S phase, then we have your G1 and your G1S cyclin. They come together and they will phosphorylate your PRB and inactivate it, and now it cannot hold your E2F, and now E2F can bind to the promoter and you can transcribe and translate your cyclin A protein. I hope students, this entire thing is clear, right? As we can see, your cell will move from your G1 to the S phase. Now let's understand about the second tumor suppressor gene product, that is your P53. Now again I will tell you, P53 is a protein. When it gets activated, it is going to block or halt your cell cycle, and this is for the benefit of the cell. Okay, don't forget. Now let's see how this P53 works. So here we have a P53 protein. Now this protein is held together by another protein component called as MDM2. Now MDM2 is a negative regulator of P53. Now what MDM2 does, once it finds any P53, it will bind to it and it will lead to the ubiquitination of P53. Now what is ubiquitination? Ubiquitination means addition of ubiquitin moieties to your P53. Ubiquitination means to add ubiquitin group to the protein molecule. Now these ubiquitin molecules are actually tagged. Now any protein who has this particular tag, it has been tagged to get destroyed with the help of a molecular organelle called as proteasome. It is a cell organelle which is having the function of degrading unwanted proteins. So how it is going to find out which protein is unwanted? It is going to check this particular tag called as your ubiquitin tag, and once it finds this ubiquitin tag in any protein, it will degrade it into pieces. Now for degrading, it is having certain proteolytic enzymes which will break down the peptide bonds between your proteins, and it will break them into individual amino acids. So now let's revise once again. We saw P53. Whenever P53 is found in your cell or if that particular protein is produced, immediately its negative regulator, that is your MDM2 protein, will bind to it. It will add your ubiquitin groups to it, and now it will tag it for destruction with the help of proteasome. So now this particular P53 will be taken to the proteasome organelle. Proteasome will recognize the tag. It will open its lid, or basically this is just an analogy to let you all know. Now P53 will be put inside your proteasome organelle. It will be now destroyed with the help of proteolytic enzymes, and it will be thrown out in the form of individual bits and pieces of amino acids. So we just saw here, now this P53 protein was important to prevent the cell cycle, but now we just saw here that there is a mechanism through which your P53 protein is getting destroyed with the help of proteasomal destruction. So now what is this role of this P53? Let us learn about the role of this P53. Now here is we have your P53, and we know once P53 is present in the cell, MDM is going to find out and it will bind to it. But if there is a signal which your MDM protein gets here, and that signal can be cellular stress. Now what is cellular stress? Cellular stress can be mitotic apparatus dysfunction, that is your mitotic apparatus. What all apparatus is required for mitosis? If they are not functioning properly, that will be called as a cellular stress. Secondly, we have something called as DNA replication stress. Third, we have telomere erosion, or the telomeres are getting broken down, or it is having any oxidative stress, or if there is any DNA damage. If there are these 5 cellular stress, they are going to signal your MDM2 to not destroy P53 by allowing the phosphorylation of P53. Let's revise till now. P53, once it is present in the cell, it will be destroyed by MDM2 with the help of proteasomal degradation. But if the cell is undergoing any cellular stress because of which it wants to stop the cell cycle, in that case it will allow the phosphorylation of P53. So your P53 molecule gets phosphorylated. Once your P53 molecule gets phosphorylated, now MDM2 cannot have a control over your P53 and your P53 is released in the form of an active molecule. Students, please take a note here that P53 protein was first inactivated, but when it got phosphorylated it became active. Now P53 is also a transcription factor. Like we saw over there that E2F was a transcription factor that PRB was holding, correct? So similarly, P53 is also a transcription factor. Now once it gets activated, it is going to give some response. Now what are the responses of this P53 can be? It can lead to growth arrest, that is it can block your cell cycle. Second, what it can do, it can allow your cell or recruit the repair, DNA repair mechanisms, so that if the DNA is damaged, it can repair the DNA. Or what it does, if it finds that your cell cannot be repaired, the DNA cannot be repaired, then it will mark the cell to apoptosis. So let's revise now. What is the function of P53? P53 protein, once it gets phosphorylated, it will prevent the movement of your cell cycle towards the next phase, or it can recruit the repair enzymes of your DNA repair system, or it can mark your cell for apoptosis. So that is the reason why, because we don't want a cell to go to apoptosis, we don't want the cell that its particular cell cycle be arrested if everything is normal. Hence, whenever P53 is present, MDM2 is going to destroy it. Now what I will do is, to make you understand better, let me just take one example. I will take an example where your DNA is damaged and how that DNA damage signal your P53 and activate it, and how that leads to the growth arrest or how it blocks the cell cycle. We will learn in detail in the next slide. Okay, now let's understand the DNA damage and the role of P53. This is your P53. As we know, once MDM finds it, it's going to mark it for degradation. But can you see here the DNA? We can see there is a double-stranded break in your DNA. It means now DNA is damaged. That is going to give a signal to your ATM. ATM is also a kinase protein. I will not discuss it here in detail about ATM. In the next video, I will be teaching you about the DNA repair pathway. So over there, I will discuss this ATM in detail. Just understand, ATM is going to receive the signal of DNA damage and it is going to pass that signal to the CHK2, which is your checkpoint kinase, and this kinase enzyme is going to add a phosphate group to your P53 and then your P53 gets activated. Okay, till then it is clear. So whenever there is a damage, there are some series of molecules, series of proteins which is going to detect the damage and they are going to phosphorylate your P53. Now P53 got activated, right? Now what it does, since I told it is a transcription factor, so again it is going to go and bind to promoter. So once it go and bind to promoter, it will allow the transcription of P21 gene. So when P21 gene gets transcribed, it is going to produce your P21 protein. Now, if you remember in our second video, we studied that P21 is a CKI, that is your CDK inhibitor. So it means now when this P21 is present, it can bind to all the active form of your CDK molecule and it can block it so that the cell cannot proceed further, right? Now here this P21 molecule is going to bind to your active form of G1-SCDK, that is a CDK molecule present at the transition between G1 and the S phase, and it is going to bind to that, and now this molecule becomes inactive. Similarly, it will also bind to the SCDK. Understand, student, here that the cell has not gone to S phase. Okay, the cell is still in the G1 phase, but before it moves to the S phase, the SCDK is synthesized and then it is formed activated because it has important functions in the S phase. So it is already activated, but now the cell get a signal that your DNA is damaged. You cannot replicate a damaged DNA. So what it will do is it will recruit your P53 molecule, make it active and allow it to transcribe the P21 molecule, and now this P21 can bind to your SCTK, and this active form of SCTK becomes inactive. Once your G1 SCTK and your SCTK becomes inactive, the cell cannot proceed from the G1 to S phase and the cell stops at the G1 phase. I hope students this is clear with you all. Thus you understood how your PRB, P53 and this P21 protein are important for blocking your cell cycle once they get activated. Okay, so let's revise it once. What we will do is we will just compare between your PRB and P53 protein and try to understand the difference. First, we understood that both are tumor suppressor genes, right? Secondly, we saw that PRB becomes active when it is unphosphorylated. It means when it is not having any phosphate, then it is active, and it becomes inactive when it gets phosphorylated, right? But if you see P53, it was active when phosphorylated, but when it was not having phosphate, it was inactive. There is a difference in both of this. Let's see here in this example. The PRB is active because it is not having any phosphate. So once your PRB becomes phosphorylated, it becomes inactive. But this is not in the case of P53 because P53 was inactive when it was not having any phosphate. But once it got your phosphate, it became active. Let's go to the next difference. We saw that PRB was phosphorylated by the active form of your DCTK as well as your ECTK, that is your G1 and your G1S cyclin, while P53 was phosphorylated by cellular stress molecules like your CHK2, that is your checkpoint kinase 2. Next, we saw that PRB is going to hold your E2F and prevent its action, while P53 is held by your MDM2 protein. Next, we understood that PRB is going to prevent the synthesis of cyclin A and block cell cycle. So the function of PRB to block cell cycle by blocking the synthesis of cyclin A, whereas your P53, how it blocks the cell cycle? It will initiate the synthesis of P21, which is a CKI, and that is going to inhibit your G1S as well as your SCTK and block your cell cycle. I hope students this was understood by all of you. This is one of the most important topic and many questions are asked in any entrance exam you face. So don't forget to revise this before going to write your exam. So thanks for watching this video. If this video was helpful, don't forget to share it to your friends, like this video and support the channel by subscribing to it. I will see you all in the next video with one more important topic in a simple manner. Thanks for watching.